Hello students, you are welcome to this study session of NSC 207. In this study session, we'll be discussing on immunity. Let me say that you have been introduced to some aspects of this course in the previous study session, and you should bear it in mind that all living things are subject to attack from disease-causing organisms. Even bacteria, so small as not more than head of a pin, can be attacked by virus, and it has its own immunity against this attack. The more complex an organism is, the more complex the immunity the organism will acquire. When you are through with this study session, you should be able to describe the components of the immune system, to explain innate immunity and cellular defense, and then you should be able to discuss specific immunity. Well, we have to discuss first the components of immune system. Immune system has some basic components, and like you can see on your screen, the diagram of the human body, which shows those areas where some cells are located to provide immunity for the body. See the tonsils and adenoid. You can see the lymph nodes dispersed across the entire body. So also lymphatic vessels. Thymus gland is found there. You see the spleen, Peyer's patches, as well as appendix. You can see also the bone marrow. All these are organs of immune system. Let me say that all cells of the immune system are initially derived from the bone marrow. In the process of hematopoiesis, that is when blood cell is being formed, bone marrow stem cells develop into either mature cells or precursor of that cells that migrates out of the bone marrow to continue their maturation elsewhere. Bone marrow produces lymphocytes, that is B and T lymphocytes, alongside the natural killers. Also produces granulocytes, and also to red blood cells and platelets. You now have cells of the immune system. The cells of the immune system are B cells, T cells, natural killer cells, macrophages, and dendritic cells. Now the B cells. The function of the B cell is to develop into antibody secreting plasma cells following stimulation by foreign antigen of bacteria, viruses, and tumor cells. Antibodies are specialized protein that physically recognize and bind to specific antigen that cause their stimulation. Antibody production and binding to foreign antigen is often critical as a means of signaling other cells to engulf, to kill, or remove that substance from the body. That phenomenon is known as opsonization. Now the T cells. The T cells are also lymphocytes, and they are divided into two parts. You have the one we call T helper cells. The same thing you call CD4 plus. That is in your HIV age, the CD4 are called helper cells, and if the virus destroys the CD4, it will reduce their number, and that will reduce the body immunity and will give room for opportunistic infection, which we have earlier on discussed. In nerve cell, you have two types of T helper cells. Second one is called cytotoxic T lymphocyte. That one destroys those cells that are tumorous in nature. The cancer cells. So both T cells can be found throughout the body, but they can be found conspicuously in lymphoid organs, like we have earlier on identified. But they can also be found on the liver, the lung, blood, and intestinal tract. The next one is natural killer cells. It can be referred to as NK cells. They are similar to CD8 plus T cells. They function as effector cells that directly kill certain tumors, such as melanomas, lymphomas, and virus-infected cells. But unlike the CD8, these cells kill their target cells without any recognition of antigen associated with these MAC molecules. So they are referred to as natural killer cells. Now cover this is the next one we are going to discuss. They are important in the regulation of immune process. Besides their roles in phagocytosis, they may function as antigen-presenting cells because they ingest foreign materials and present these antigens to other cells of the immune system, such as T and B cells. This is one of the important first steps in the initiation of an immunological response. So what this one does is that they will just ensure that they engulf the microorganism, they ingest them, thereby exposing them to the action of B and T lymphocytes. Dendritic cells also originate in the bone marrow and function as antigen-presenting cells. In fact, these cells are more efficient than macrophages. These cells are usually found in structural compartments of the lymphoid organs, like we have earlier on mentioned. They are also found in the bloodstream and body tissues. It is believed that they capture and process antigen in lymphoid organs, where an immunological response is initiated. It may interest you that current findings have shown that these cells bind large number of HIV particles and may be a reservoir of virus that is transmitted to CD4 plus T cells. Those are the cells of immune system. They are also represented in this diagram. You could also see those cells. You also see how they are dispersed around the body. You have the macrophages, you have the mast cell, platelets are there. These are dendritic cells, like you are familiar with in your neuron tissues. The B lymphocytes, natural killer cells, T lymphocytes, and the lymphoid progenitor cells. They are all the cells of the immune system. Now we discuss innate immunity. When you say immunity is innate, what does that stand for? It means it's a form of non-specific host defense against invading pathogens. It is natural or innate to the host, depending in part on genetics. That is, innate defense mechanisms are constituents of the host. They are continually ready to respond to infection and not require a period of time for induction. It means one is born with it. It contains some specific, what we call first line of defense mechanism, like the skin and mucous membrane. The skin is a first line because all organisms that want to enter the body must pass through the skin. And intact, unbroken skin is a good physical barrier to infection. Let me say that also in the skin, you have secretions of some sweat, and that sweat contains some chemical components that are destructive to microorganisms. Apart from that, you have some mucous membrane, like the one lining the mouth, on the wall of GIT. All of them have one property or the other that will prevent these pathogens from invading the body. If they invade the body, they could not cause any harm to the body. So they are called non-specific defense mechanism simply because they can work against all forms of microorganisms. To talk of innate immunity, there are two types. We have what they call non-cellular type and we have cellular type. Non-cellular type and cellular types. Some of them are cellular in nature, while none of them are non-cellular in nature. What you mean by this is that we have discussed some cellular defense mechanisms. Then we talk of BT, natural killers, macrophages, and dendritic cells. They are called cellular defenses. There are some that are not cellular defense, but they are still referred to as anatomical defenses because when they maintain their integrity, they serve as immunity to all forms of microbial infection. You will see here the non-cellular defenses in this diagram. Like I told you, the eyes produce the tears, and these tears have both chemical and mechanical nature of destroying microorganisms. Also, respiratory tract has some, GIT has some, always, and also your urinary tract. They also have protective mechanism. Cellular defense may be inflammation and phagocytosis. Inflammation is telling you that when the antigen infects the body, it will initiate some reactions, and these reactions will bring out a kind of defense against these microorganisms, and we just manifested in terms of signs and symptoms that shows that there is another antigen that entered the body, in terms of cardinal signs and symptoms of inflammation, which are redness due to increased blood flow, swelling due to increased vascular fluid escape to that area, and phagocytic infiltration to damaged area. The heat is also one of the clinical signs due to increased blood flow to that area, and the pain is also there because some tissues there are destroyed. These are what we call classical signs of inflammation. When you talk of phagocytosis, if inflammation cannot do the work, then the cellular part of the body will now ingest these microorganisms, now exposing them to the destruction by the cells we have earlier on mentioned. And this is phagocytic process, like you have it, that this microorganism will open the mouth and will ingest the microorganism. And when they get to the facule, that is where they will be killed, digested by these T cells and B cells we have mentioned earlier on. This is the process of phagocytic action. And then we talk of specific immunity. This immunity are specific because they can only work against a particular microorganisms. They are not as general as non-specific. Two types of white blood cells are known in this category. The lymphocytes is what we call, and then they are produced in bone marrow and mature into one of several subtypes. The most common type we have mentioned are T and B lymphocytes. They are specific immunity because they can only fight against specific microorganisms. You now have what we call active and passive immunity. When you talk of active immunity, animal undergoes an medical response to an antigen and produces the cells and factors responsible for the immunity. Animal produces its own antibodies and or immunoreactive lymphocytes. Active immunity can persist a long time in animal, up to many years in human, because it is active, because the infection of the antigen actually initiated its production and they are found in the body. But they could be given as exogenous route, because you can give an injection that will stimulate this immunity and they are still active, because it is not that injection that will fight the infection. It is the injection that will just stimulate those antibodies in your body and will fight against the infection. We call them active immunity. But when you talk of passive immunity, this one is a process immunity from animal which are produced in another animal. So instead for the body to produce the antibody, that antibody is now injected into that individual and will now work as antibody against that invading organisms. So that makes the difference. If you talk of active one, it means the body stimulates its own antibodies by itself, but can be assisted, can be initiated. But in passive immunity, the body is not stimulating or is not synthesizing any antibody, but instead of that, that antibody, a prepared one, synthetic one, is now given to that individual so that it will help in producing, it will help in fighting against that antigen that entered the body. Let me give you an example before I leave here. When you give somebody this, I will come to you now with a fresh wound. You will say, give ATS and give TT. When you give tetanus toxoid, you are just trying to initiate antibodies, natural one from the body, to fight against tetanus. When you give ATS, it is serum of other animal, so which is given to fight tetanus. So it is not, that ATS is passive, while TT is active. Students, I believe you have learned much from this session. When next we meet, we shall discuss more interesting topic on your learning program, NSC 207. Until then, thank you for listening.